So this is the first half of a presentation on synaptic communication. The first one we get into is all about kind of the types of synapses that are in the central nervous system. So the idea here is we have two major synapses, two major types. We have electrical synapses and we have chemical synapses. Basically, it's the idea of the soups versus sparks theory of communication. Soup meaning kind of like your circulatory system. The nervous system is just kind of one big flowing thing and everything just is connected to each other directly. The sparks theory is that we have the action potentials that are transferred from one cell to the next through some kind of synaptic transmission. So we'll kind of look at the difference between those two things today. So for some basic terminology, we have the dendrite. That's the region of the neuron that receives the input. So for this cell that's going to be down here, there's our dendrite. Okay, it's going to receive input from the axon, which is the output part up here of the previous neuron. There are some times where the cell will actually synapse back on itself, so it may be the same neuron. But for what we're doing today, we have a dendrite and we have an axon. We also have what's called the presynaptic cell. So that is the side that's presynaptically. That is our axon, is the presynaptic cell. You also hear me refer to the postsynaptic cell. That's going to be down here. Okay, that's your cell on the other side of the synapse. So we have dendrites, which are going to take in information, axons, which are the output portion of the neuron that relay the signal. Presynaptic cell and postsynaptic cell kind of correspond to those a lot of times. But again, sometimes your synapses are going to be on other parts of the neuron, which we'll talk about in a little bit. But to start with, we're going to talk about electrical synapses. These are also what is known as gap junctions. They're formed by 6 connection subunits. Okay. And they're going to cause cells to be what's called electrically coupled, meaning whatever happens in one is going to immediately happen in the next. So it's going to allow for very coordinated activity between multiple cells really quickly. Ions are going to flow directly through the cytoplasm. Okay, there's no extracellular space where things are going to float around in. Basically, the cells are going to be connected. The flow can be bidirectional, meaning the ions go one way or the other, and it's almost instantaneous transmission. So what does that kind of look like? So right here and right here where those arrows are pointing to, you will see some gap junctions. The one thing you'll notice, say in the cell on the right, is that in the cells on the right, even though there are a lot of vesicles and a lot of neurotransmitters floating around in this cell, right around that gap junction, you don't see any neurotransmitters. So that tells you that there are no vesicles being released at that location. On the left-hand side, you see there are no vesicles at all in either of those two cells, but there is an electrical connection which they kind of zoomed in on, and you kind of see these little dense regions right here. Okay, so this is an electron micrograph, so that's electron microscopy, meaning we're getting down on really tiny scale, obviously. We see in the nanometer range, and we see these synaptic zones that are definitely not chemically based. So here's kind of a zoom in view of that. You see our gap junctions in the membrane here, and you have the channels, the connexins actually make their way from one cell to the next, and they end up with that gap between the two where ions can move back and forth. So each cell is going to have to develop its own connexins, and those connexins together on each side will bind together and form your gap junction. So here's kind of an example of what it might look like electrically recording. So this is some tissue suffering from seizures. The action potentials were forming in random bursts, which you can kind of see right here, kind of zoomed in on it right here. So there's a baseline recording, meaning just whatever is happening naturally. You can see there's a whole bunch of clusters of signals, okay, of action potentials being developed. Okay, some kind of extreme levels too. However, they put on this carboxylone, okay, and you can see as the concentration gets higher and higher, it's been soaking in there, those bursts are now gone. So now you see nothing, okay, just basically stays at baseline. So basically we've eliminated the electrical connection between these two cells, or between the set of cells that is reducing the seizure-like activity, the coupled grouping activity between those cells. We wash it back out, meaning basically we just put it back in normal solution. And again, you see the clusters of activity come back. And what they just want to show down here is that they can still elicit action potentials. So we're not actually blocking action potentials at the individual cell. We are blocking these clustered groups of activity, these random bursts that show that are kind of seizure-inducing type things, where just fits of electrical activity that are coordinated. So this gets into the idea that gap junctions are going to be used for coordinated electroactivity. You think about places that might be useful. Well, one is your heart. You gotta get a lot of muscles to work together at the same time. Your digestive system, okay, you want to make sure it's moving things along, okay. And there's other places too where gap junctions will play a role, but the main method of communication for the central nervous system are chemical synapses. So again, the synapses have their pre and postsynaptic site. They're not bidirectional, they're one directional. That synaptic cleft, which is this gap down here between the two cells, it's only about 20 to 50 nanometers wide, kind of extremely small distance between these two cells, but there is a gap between them. The axon terminal is going to be loaded with vesicles and mitochondria, going to have both those two things. And the postsynaptic site is going to have receptors for receiving signals. So that's what these little dots are down here. They're receptors of some kind, depending on what type of cell it is, that's going to receive whatever the signal is coming from that presynaptic cell. Now here's kind of an electron micrograph of the same thing. So we're zooming in really fine again. You can see the heavy amounts of mitochondria, okay, telling you that synaptic transmission is extremely energy dependent. Okay. And a whole lot of vesicles stored all over the place. There's vesicles everywhere. Okay. You have kind of some standard vesicles. You also have these things called dense core vesicles, which will usually have a different kind of neurotransmitter in them. I'll talk a little bit more about that in the next video when we talk about neurotransmitters. But we have basically a ton of vesicles, a ton of mitochondria, and on the other side you'll see nothing. There's no vesicles whatsoever on the other side of the membrane for the postsynaptic cell, and really not much mitochondria either. It's not a very active process on the other side. The other key thing that you will see though are these active zones. So if I highlight them here and here, those active zones are the place where synaptic transmission actually takes place. So, whoops, I didn't want to write in white, I wanted to erase. So that's where the vesicles are actually going to fuse, bind, and get released to the postsynaptic side. Okay? So it doesn't happen everywhere along the axon terminal. It happens in very specific locations. Okay? And that's important because you want to understand that when we get released to the other side, it may only be from one of like 10 or 12 different synaptic active zones within a single synapse. Okay? That means that there's other places where there might be vesicles ready to go for the next time a signal comes. Okay? So moving on to types of synapses, we have 3 different types of synapses that we're going to deal with here in terms of where the location is. So that's one way of classifying like what type of synapse it is. And the first one is called an axodendritic synapse. That's pretty simple. That's kind of our standard, what you would imagine. The axon is synapsing right here on the dendrite of the next cell. Okay, so we talked about that being dendrites kind of being the gatherer of information, bringing it into the soma. So this cell here is influencing the information that's coming into the soma, or generating that information coming into the soma. You may have axosomatic synapses, okay, where the axon is going to synapse on the cell body. Okay, that's going to, because of the volume of the cell body in the soma, you're talking about probably having a smaller effect on the outcome for the postsynaptic cell, but probably some kind of regulatory effect of what is that postsynaptic cell going to do with all the incoming information, right? So you've got all the information coming in from the dendrites, and then you might be able to slightly regulate that by influencing what's happening in the soma with an axosomatic synapse. And then you might also have axoaxonic synapses, or axon to axon. So this cell right here might shut off. So this cell might be firing action potentials, okay, and they're coming down its synapse, but then this other cell over here fires an action potential that actually blocks or shunts that signal coming in, and basically cuts off the activity of this cell onto the other one, or maybe increases the level of activity from that cell onto the other one. So you can influence the outcome of the postsynaptic cell in multiple different ways via synapses. And it all depends upon what type of neurotransmitters are there and what's at the junction. So one of the most highly classified synapses is the neuromuscular junction. It's very highly characterized, and you just establish a lot of the basic principles of what's going on with synaptic transmission. Basically, that's the junction between a motor neuron sending its axon down and making contact with a muscle fiber. They're very stereotyped. They're very easy to pick up. They're very easy to record from the output when you're recording from the muscle cells. So they've been very highly characterized and used a lot to figure out what exactly is going on in synapses. So here's kind of an example of the neuromuscular junction zoomed in a little bit. You can see over here with the immunocytochemistry, you can see that nerve come in from the outside, and then it branches off into all these different things here. So where this intersects, this is called the motor end plate region. So this is called the end plate. Okay. And it's very organized. It's actually very stereotyped, even from animal to animal. They're going to tend to be towards the middle of a set of muscle fibers is where you'll find these neuromuscular junctions. So if we zoomed out on this muscle, it might look like this. Here's your entire muscle, and then your neuromuscular junctions will almost be lined up right down the middle of it. Okay, with that incoming nerve fiber going like this. Okay. Which makes sense because that's where you want to stimulate your response, is in the middle, and you need it to be very stereotyped. So that way it's easier to maintain consistency for muscle movement. So if we zoom in even more though, they did the electron micrographs in neuromuscular junction. You can see that you actually have these folds in the junction, okay? These folds right here, and you can see them over here in the micrograph too. Okay, place where basically you can ensure that you have more receptors to receive the neurotransmitter that's being released from the presynaptic cell. So here you get the neurotransmitter. It's going to come out. It's going to bind down to here. The neurotransmitter in this case is called acetylcholine. And we will talk more about all the neurotransmitters in the next video. But the acetylcholine is going to get released from those vesicles in the presynaptic cell, and all that acetylcholine is going to start to bind to these postsynaptic receptors down here, trying to activate the cell. Now, what's going to happen electrically, here we see some recordings at the neuromuscular junction with both evoked and random responses. So the EPP it's referring to here is what's called the end plate potential. Basically, they're just measuring the memory potential of the muscle fiber. And then you can see you get like a 20 millivolt change when you give a stimulated response. So basically you activate that nerve muscle fiber that's coming in, and you get this large end plate potential. Now you can imagine if you get multiples of these end plate potentials, so multiple responses coming in, okay, and they sum together, you're eventually going to cross threshold and you're going to develop the action potential in the muscle fiber, and you get the muscle to contract. However, what they also notice is something kind of cool, is there are these mini end plate potentials that they see just spontaneously as they're recording. See, they're really small. They're only like, what, 1 millivolt compared to the 20 millivolt one over here. So that tells you that it's not always just coordinated release happening. These vesicles are being released randomly too. So it is the coordination of that release that's the key to generating that postsynaptic response in the cell. So kind of some basics here of what's going to happen as the action potential travels down the axon. Okay, it's going to make its way into the postsynaptic cell. Then you have voltage-gated calcium channels open. That's these guys here. So as that voltage comes down, it opens up these calcium channels. That causes calcium to flood into the cell. Okay, and that calcium that goes into the cell is actually going to increase internal calcium. So there's little internal stores of calcium inside, And as that internal calcium is released, it's going to cause those vesicles, kind of shows in the diagram, but I'm going to draw it anyway, to fuse with the membrane. And when they fuse with the membrane, they get caught in like this thing called a snare protein. Okay, through what are called snare proteins. Okay, they fuse to the membrane and then they release their neurotransmitter into the postsynaptics, into the synaptic cleft. There, it's going to bind to those postsynaptic receptors and do whatever it does. We don't know. It all depends on what these types of receptors are. In this case, it says sodium channels, but it could be any. There's so many different types of receptors and different types of neurotransmitters, which you'll find out about. In this case, they mentioned it's sodium, but it can be lots of different things. And then the neurotransmitter is there, and then eventually it's going to be removed from that cleft. So it's going to vacate the cleft either through diffusion or recycling, maybe get taken back up into the previous cell, or it's going to get degraded via esterases. So lots of different things can happen once that neurotransmitter gets released. But that's kind of the basics for neurotransmitters. So again, basis for synaptic transmission. So again, you have neurotransmitter synthesis. You need to make the neurotransmitters. They're going to get packaged into vesicles. Those vesicles with the right signal will fuse to the presynaptic terminal. Then the neurotransmitter is going to spill into the synaptic cleft. It's going to bind to the receptors after it does that. You're going to get some kind of response. We'll talk more about that in the next video, like what types of different responses there might be. And then you're going to have to remove that neurotransmitter from the synaptic cleft. And that'll happen through multitude of ways too, depending on what the neurotransmitter is and what synapse it is. So hopefully this gives you a good outline into the basics of the two types of synapses. And then we'll talk more about the details of those neurotransmitters and what their postsynaptic responses are in the next video.